Right, as you can maybe tell from the pictures on here, I work in an institute, the Sanger in the Tree of Life, where we sequence lots of eukaryotes, or at least that's what we attempt to sequence. And because the samples that we use are caught from the wild, typically, sometimes there'll be other genomes mixed in there from organisms that are associated with the target. And sometimes those can come from groups that we haven't actually been able to sequence very extensively before, such as myxozoans, which I'm going to talk about. So I'm going to be talking about this in more of a context of a case study, so there's not going to be any benchmarks, but I'm interested in how embeddings of different types of the sequences, either of scaffolded sequences that have been assembled or the raw reads, might be useful to retrieve genomes that could be interesting but are not easy to get. This isn't doing anything. Can I use the keys?
See, you should be able to, but there. Just slide.
Oh, I was pressing the wrong way. I think, yeah. Right. So two of the fish assemblies that came through the pipeline at the Sanger had some strange sequences in them that did not seem to belong to the fish. So we can look at some tetranucleotide projections in 2 dimensions of the sequences. And you can see there's a cluster to the left of sequences that don't have any blast hits, and they compositionally don't appear to resemble the scaffolds that have been labeled as fish. So there's 2 cases where this occurred. And the only sort of hint as to what might be in this cluster is that there are some ribosomal sequences that match Kudoa, which is a myxozoan parasite that happens to infect marine fish. So the question was whether we can work out whether the other sequences in that cluster, because the composition is kind of all over the place, come from the same source. And one way this is routinely done in curation is to look at the chromatin contacts. So you expect that if something belongs to the same genome, it should be physically interacting. So the curators will look at these nice maps and try to puzzle together the chromosomes of the target and then remove anything that hasn't been caught by another pipeline to get rid of it. But the issue with that is you need to be an expert at sort of interpreting these. I certainly wouldn't be able to do it. And sometimes there are sequences in there that are too small and they get missed. So after I got my hands on this assembly, they ended up having to re-release it because I found some things that were left over that shouldn't have been in there. So you can see here there's some fish chromosomes and there's some not fish. So I was interested in seeing whether, hang on. No. Sorry, I have left-right confusion. Right, I was interested in seeing if there's a simpler way of sort of pulling these things apart that doesn't require any skill that even I could apply. So instead of looking at these elaborate maps, I just asked for each pair of scaffolds, is there evidence of a connection? And I put that into a matrix. So you might imagine it would look something like the diagram here. If you had 2 components in the sample, and then I stuck that into a graph and embedded the nodes of the graph. So the nodes are the scaffolds in this case, generated some embeddings, and then attempted to cluster those. And if that clustering is successful, and this is unsupervised, by the way, you ought to then be able to pull apart the 2 separate genomes. And the method that I used to group the scaffolds in the graph is something called node-to-vec. I'm not doing this deliberately, I promise. And node-to-vec lets you detect groups of interconnected scaffolds. And the way that it does that is it wanders around the graph and samples nodes, and it tries to give neighbors in the network similar vector embeddings. And there are 2 sort of broad notions of similarity or neighborhood. One is that different nodes could have similar structural rows, so they might be hubs or something. But what I'm really interested in here is finding communities. So in order to bias the random walk to favor that sort of setup, we can tweak some parameters. So if we're at node V, having just arrived from node T, there are three options. We can backtrack, go back to T. We can stay in the local neighborhood and go somewhere that's also connected to the previous node T, or, which is the thing that's appropriate for this setting, we can bias the walk to venture somewhere else, further out. So I set the parameters accordingly to slightly favor the latter of those. In practice, I found that different parameters kind of did the same thing, and that might be to do with the structure of the network. Oops. So the figure at the top is what my adjacencies looked like before I tried to put them through the model. Anything that's shown in white indicates that there's at least one connection between that pair of scaffolds. And after this has been through node-to-vec and clustered with k-means, we can see there are two distinct components. So at the bottom, you can see the sorted version of that matrix. And importantly, the cluster that was previously unassigned is the same cluster that contains the ribosomal subunits. And we can see those labels applied to that previous plot here. The only outlier is that the mitochondrion, it's a mitochondrion, so it's compositionally not that similar. So the other fish looks the same as this. So we've got one fish, one myxozoan each. And why is that interesting? What is a myxozoan? So they look like this. They're quite simple. They're obligate endoparasites. And all they kind of do is infect one of their hosts, swim around, infect the other host. And what's particularly interesting about these is that they may not look like it, but they're actually animals, they're cnidarians. So they share an ancestor with these jellies and anemones and corals. And you can see them, hopefully, within the cnidarian tree at the top. And the two that I pulled out are highlighted in red. So in addition to this morphological simplification that you could see on the previous slide, the genomes have really shrunken as well. Not only are they small, they've gotten rid of upwards of 2/3 of their genes in the case of Cadoa, but we don't know particularly much about the structure of the nuclear genomes because there aren't that many long read assemblies for them. There's one, but I don't think it's been scaffolded. So it would be really convenient if by accident we'd kind of managed to sequence two of them. Unfortunately, it seems as those preliminary scaffolds that were pulled out of the fish don't have the entire genomes. So at the top, we've got the closest reference that's been released. And you can see that both of these assemblies are a bit on the small side. And that doesn't seem to be explained by the contiguity, which is higher than before, which you'd expect from the technology. One of them in particular also appears to be missing a bunch of genes. So given this, I wanted to try to go back and reassemble these correctly and try to get the whole genomes. But before I did that, I wanted to get an idea of what I was dealing with and figure out roughly how much there was in the raw read sets. So we saw before that there's compositional differences at the scaffold level between the fish and the myxozoan. And we expect to see the same at the level of the reads. So what I did was visualize the data set using a variation autoencoder, which is exactly what I'd done to the scaffolds, but it was a bit more appropriate here because it, unlike UMAP, which was referenced earlier, you can stick millions of sequences in without it sort of running out of memory and crashing. So the inputs, you've got a summary of the sequence composition in the form of tetranucleotide counts. Those go into an encoder network, and that projects the sequences down into a two-dimensional space. And the latent variable Z is a sample from a normal distribution based on some parameters that are given by the encoder. Z then goes into the decoder, which attempts to reconstruct the initial inputs as well as it can. So the objective is to attempt to minimize the reconstruction error as well as keep the latent space close to the Gaussian prior, so you get a compact representation. So the intuition is, if the information that's encoded into Z is useful and has salient information about the higher dimensional space, sequences that are similar to begin with ought to be close together in Z. And in practice, to achieve that, you have to tune down the regularization term. So this is actually a beta VAE. And this is what happens when you apply it to one of the fish data sets. You can see there's different splotches. Part of that is just because the fish is heterogeneous, but the splotch in orange has the reads that are mapped to the previous Cadoa scaffolds in it. And the reason that I can't use PCA is that the variational autoencoder is just better at this task. Right, so I haven't fully answered the question of how much parasite is in here. But what we can do is, for some substrings of length 31, you can count how often those occur across the entire data set and average that across the read and draw that onto the latents. And as you can see from the dark color, there's not that much CUDA in comparison in here. You can then sort of pluck that region out, give it a little histogram by itself, and it's in the region where normally you'd be throwing out the data because it's full of errors. But in this case, it's actually interesting. So this is somewhat of a problem because the assembler might be prone to attempting to get rid of these sequences and it might not get the whole genome out. We tried to do more sequencing. We ended up with a bunch more fish reads mostly. But fortunately, in this case, because these were hi-fi reads, a simple meta assembler actually did a good job of assembling this, and I was able to scaffold it. The other sample was a bit more uncooperative because the read quality was slightly lower because it was different technology. So when I attempted to meta-assemble it, it just crashed. But I was able to hack this problem and get around it by extracting the region of the latent space where I expected most of the Codera reads to be, put it into a simpler assembler, and that worked out. And if we compare these to the previous assemblies that are shown in gray, we can see there's improvements in the size and contiguity and the completeness. And the size of the smaller one is about what you'd expect from the K histogram. The larger one is kind of interesting because if you look, the assembly size is almost the same as a scaffold N50. And what that means in practice is there was one very big scaffold that has almost all of the genome, which suggests that there's one chromosome in this. And if you show this to somebody who knows how to interpret these maps, they will say that this looks like one chromosome. So we've now got these two genomes that seem serviceable, and we can ask some questions about evolution. So we can ask, how does parasitism affect how the sequences evolve? How does gene order evolve, given that we have most of the chromosome in at least one of these? Now, in free-living cnidarians, we know that over time, the contents of a given linkage group, so a chromosome, tend to be the same, but the order within that chromosome gets scrambled over time. So you can see a more recent split, a slightly older one, and the more recent one has got a similar diogenes of the two Cadoas. So it's a bit of a surprise, given all the turnover that's happened in the ancestry of these, that these seem to be remarkably collinear. There's not that many rearrangements between them. And you could speculate as to whether this is because if they rearrange anything else, they're going to seriously break something, or it's because they don't have an opportunity to do much rearrangement because maybe they don't recombine. I think people are still speculating to the extent to which they actually do outcrossing. The other really obvious thing to look at is their rates of protein evolution. So you might note that the branches at the top of the tree are very long in comparison to the others. So these are known to have very fast evolving proteins. And one naive interpretation of that could be that because they're parasites, they need to co-evolve with the host and keep up and sort of adapt to their niche. Another is, because of their life cycle, which has got two hosts, they get bottlenecked over and over again. So that might increase the rate of drift and allow them to fix more deleterious substitutions. So I wanted to see if I could find a difference in constraint between the myxozoan and the free-living branch, but it turns out they're actually too far apart. And if you try to do that, you get bizarre values. And you can sort of diagnose this by looking at the synonymous divergence. Basically, anything in the histogram that goes into the red range is considered too diverged to render any sort of reliable estimate. But you'll see that there are two regions of the tree that are sort of in the blue zone. So we can look at the branches that are a bit shorter and then ask this question again. And what I found is that there's a slight acceleration in Kudoa, and somehow I've got a line break in my axis label, which is on, yeah, it's on the mislabeled one. But this is in principle consistent with less effective selection, but it's maybe not as staggering as you might expect. And that could actually also be to do with the fact that these are still relatively long branches. So it's known that when amino acid preferences in proteins are site heterogeneous, you try to compare a pair that's very diverged, you will tend to underestimate the non-synonymous divergence and therefore omega as well. And yeah, there's all sorts of other things going on here. There are deletions, and we don't typically account for those in our models. So trying to interpret this, what do I think is going on in myxozoan genomes? Are they streamlined to be really good parasites that have become really efficient by getting rid of genes that they don't require? Or are they just drifting around and deteriorating and losing things that are maybe actually useful? So some people have attempted to see if there's evidence of positive selection in some of these. I tend not to believe any of those results because of the aforementioned difficulties just getting simpler rate estimates. The other thing, is that there seem to be deletions that are still ongoing in the Kudoa tips, and you could speculate whether that might also be deleterious. And their genomes may be small, but for something that's supposed to be streamlined, they have quite a high repeat content. So that's not necessarily what you'd expect if selection were driving the genome contraction. And in Kudoa, gene order is really static, but it's not clear that that's to do with any sort of selection, because there are other groups within myxozoans where if you look at the mitochondria, they're incredibly scrambled to the point where you can't make proper trees with them. So in summary, these things are really weird. Their genomes are really weird. Unsupervised learning can be used in conjunction with more traditional assembly methods to try to dredge out some useful genomes. This could also be applied, of course, to other undersampled taxa. But when it comes to trying to assess what's going on with sort of selective constraints in this lineage, we actually just still need more sequencing to be done. And some of it could also be done deliberately, not by accident. And in the meantime, I think I don't believe anyone who says anything about positive selection. And there are various people that I've had discussions with while doing this work. And the code for all of the things that I talked about, so the variational autoencoders, the nodes of EC, are on my GitHub.